now set up and we're ready to begin. I'd like to welcome you all to this week's BIMR seminar. We are starting a new theme this week for the next 4 seminars. The new theme is on optical microscopy techniques, high resolution microscopy. And today we kick things off with Dr. Joao Frimino, who is the facility manager for the Center for Advanced Light Microscopy here in this building at McMaster. So Joao did his undergraduate studies at the University of Lisbon on microbial biology and genetics. He, as part of that, did an internship in Cambridge and followed up his undergraduate studies with a PhD at the Max Planck Institute for Molecular and Cell Biology in Dresden, Germany. After that, he did a couple of postdocs at the Pasteur Institute in France, and the second one in Lausanne, Switzerland. And in 2019, he became the facility manager for the microscopy facility at the University of Toronto in the Department of Dentistry, facility called Analog. He was there until 2021 when we were fortunate enough to attract him here to McMaster to essentially start and set up and launch the Center for Advanced Light Microscopy, which we're very proud of, and has been growing thanks in large part to Joao and his efforts. He's been setting up the equipment, training users, making sure the facility runs and operates. And today he's going to tell us about how this new facility helps support materials research here at McMaster. So with that, I'll turn it over to you.
Thank you very much, Alex. Appreciate it. Thanks for the opportunity from the BIMR to take over some of your seminar symposiums so we can actually also spread the word on light microscopy and what can be done for research here at McMaster. I would just like to add from Alex's kind description of what we do that this is also a team effort. It's not just me. There's a lot of people involved with COM, and this is why this is a successful endeavor so far. So to get things started, just want to make sure that everyone by the Zoom link can listen to me. Can we get a thumbs up? Everything's okay? And everyone in the auditorium can also listen to me? Perfect. First things first. So everything you're going to see in the seminar can be photographed, tweeted. You can share the information here. There's no problem whatsoever. Feel free to do so. There's no unpublished data or data that is sensitive, so feel free to do so. And without further ado, let's just start the presentation. So the goal today is actually to give you a flavor of what COM can do. We'll try to focus this more on material science because this is what the BIMR is mostly focused about. But we can do also many more things than just materials. We can do biology, we can do clinical stuff, we can do biophysics, we can do many things with the instrumentation that we have downstairs. Again, if there are any questions, do not hesitate to raise your arm. This is going to be very interactive, so feel free to intervene whenever you feel the need to. So COM started from the initiative from 3 different faculties at McMaster. The Faculty of Science, Faculty of Health Sciences, and Faculty of Engineering thought that it would be important to pool the resources and create a light microscopy facility that was lacking in the research environment here at McMaster. And that's how the idea initially started and blossomed. And since the inception of the idea until the renovation, there was a long period of time, as Jose can definitely attest to that. But now we are finally here. Our main purpose is to be a hub of excellence in light microscopy and not only provide instrumentation, but also support, scientific support to all the researchers at McMaster and also outside of McMaster. We're not just exclusive. Also, we will help to create a new generation of microscopists. So that's something that we aim to do also on the educational side. Not everyone that goes into the facility can become or wishes to become a microscopist, but if there is will, there might be a way to turn you into a microscopist by using our services. Quick highlights of the first year of operations. We have trained more than 150 users so far. The space, it's basically 3,000 square feet in the basement of EBB. I don't know if everyone in the auditorium or in the Zoom has been there, but it's a very nice space. Feedback has been quite good about that. Also, one advantage that we are very close to all core facilities in the basement. We are close to CCM. Basically, it's a 20-step trip from our facility to their space. We're also close to the X-ray facility, to the crystallography and MR. Basically, we are very well located in terms of supporting the other core facilities. And as I said in the beginning, this is a user-oriented facility. You can theoretically be there 24 hours, 7 days of the week, if you so desire. I will not recommend it. That's possible if you do wish to do so. So as I said, we are relatively young in terms of when we actually open doors to our users, but the idea was actually much more established in the past. So officially, we were established in April 2021. This is when the renovations were about to finish. And we opened the doors to our users in August 2021. Again, initially with a very small batch of users, so test subjects, let's call it this way. So we can really test and stress our workflows that we are going to establish at COM. And to give you an idea about the governance structure, so as I said, we are represented by 3 different faculties: science, engineering, and health sciences, and we have 3 directors for that. So Professor Jose Mirabal is representing the Faculty of Science. Professor Qin Fang is also representing the Faculty of Engineering, and Professor Truant from the Faculty of Health Sciences. It's a triumvirate, but again, acting director currently is Professor Jose Mirabal. This is a rotating cycle, so there'll be a time where it'll be engineering and then eventually health sciences. In the facility at the moment, on the terrain, there's me. There's also Dr. Mohan Babi, who is in the audience. Most of you might have known him or seen him along the corridors at ABB. And basically, we have a lot of experience in biology, in super resolution, even in material science. We have a lot of expertise and many years of experience combined. Finally, we also have advisory committees to basically see the evolution of how the facility is progressing over time. We also created user and infrastructure management work groups. So we always want to get feedback from the users and from people from within the infrastructure at McMaster, so we can improve and get better and be more efficient, and so on and so forth. We also have an equity, diversity and inclusion work group, and we also have outreach work groups that we want to establish very soon. So how are we supposed to help promote research at McMaster? As I said, having a nice work environment space is important. Having good instrumentation is important, but also having support at every step of the way of the scientific process is also extremely important. So one thing that we pride ourselves in is that, let's say you come up with a scientific question and you have some ideas of how you want to address it, and you think microscopy might be a good way to do so. You can come to us, you can explain what you want to do, and we will be happy to advise you on how to prepare your sample, or which way we can mount the structure you want to image, or which technique might be more suited to get the data that you require for your publication. So we have a consultation step where we exchange ideas, we learn. We're also learning from our users. It's not a one-way street. We learn from what they want to do. We think of ways that they can help their workflow, and hopefully they can also learn from us in terms of tips, tricks to maximize their output. So then again, once the users have samples and they are properly prepared and suited for imaging, we help them either by training, so we can train our users with the microscopes. And again, our training is quite, I'll not say unique, but it's actually quite thorough, because we want you to understand the machine that you're going to be using. It's good that you understand how the instrumentation works, so you can actually understand the data that it is producing. And again, we also offer service of imaging. Let's say that you don't have the time to learn microscopy, you just want to get a quick figure for your paper. We can also be used as a service. We can image for you if you tell us exactly what you need. Again, then we can also help you through the step of image processing. Again, you're going to acquire data. These days you have to quantify. You cannot just show nice pictures anymore. Those days are long gone. And we can establish workflows. We can write macros. We can write image processing routines that will automate your analysis and get your results faster that way. You don't have to basically count cells one after the one manually anymore. This can be done automatically, for example. As I said, so then we have custom-made macros that we can write. This is going to basically extract information required from your images. Then you can actually process this data. And again, we can help you with this data representation. And hopefully by the end, you can have data that you can actually publish and move on with the next project and so on. So again, all this can be done within COM. From the sample preparation, we have spaces where you can actually sample prepare. We have instrumentation, and we also have image analysis rooms where you can actually have workstation power to actually process all this data. For those of you that are familiar, we are located in the basement of ABB. This is a plan of the facility. So we have a main imaging room with most instrumentation. We have basically wide fields, optical computerized tomography systems, 3 confocals, 1 laser microdissection system, 1 AFM there, 1 EVOS, which is a multi-plate reader. We also have a SIM for super resolution structural image microscopy. And very recently, we also got our confocal for cryo workflows, which is, I'll say next week is going to be fully operational, so we can actually start training people on that system and its capabilities. We also have an image analysis room, as you can see there. And most importantly, we have a cell culture space where you can do your cell culture if need be. Or if you're more into materials, you can also eventually, not yet, this is still work in progress, but eventually the microfabrication lab will be operational, and you can actually produce and 3D print structures for your research if you need to do so. I also have to say that we have a storage space where you can keep your reagents. Excuse me. Apologies to people at the Zoom. So we have basically a storage area where you can keep your reagents for whenever you want to do your experiments and manipulate them. And we have 2 offsites, one offsite at FHS, where we have a small instrument there, an EVOS for live imaging, and 1 Asylum AFM at engineering, in the basement of engineering, but this is not highlighted here. So this is pretty much what I just said. So we have a sample storage room with a -80, a freezer, cabinet space where you can keep your reagents. In the microscope room, we have 3 confocals, 1 AFM, 1 microdissection, OCT, wide field EVOS. Very soon, meaning in a month's time, we'll receive our cell discoverer. This was a system that we demoed this past year, which is going to be very useful for people that do multi-well imaging. It's a fantastic system. It's really the first in Ontario at the moment. And towards the end of the year, we'll also receive a slide scanner for your volume imaging. If you want to image many slides, you can also do that in the cell, in a slide scanner that we receive. And then we also have the super resolution systems. And I forgot on the previous slide that we also have a station on the 2nd floor here, where you can do storm and turf if need be. Again, I'm not going into the details of what these techniques, but you can come downstairs and we can actually discuss what these things can do. Again, as I said, we also have the cryo microscope that recently arrived, the microfabrication room, which will open next year. And in the cell culture room, we have 2 BSCs, fridges, incubators for cell culture work. We also have a nice bench space if you want to prepare samples in there, and the image analysis room. One thing that also is quite unique about COM is that we took and saw the potential in establishing a cell line repository for people that do biological work. So we are starting to collect multiple cell lines in this repository, which people can just ask us if they want to test a different cell type, they can just ask us. And if you have it, we'll gladly share with you at no cost whatsoever. And I said the offsites too. So the images represent different areas. This is the workstation area. The top one is the BSC in the cell culture lab. Okay, let's talk now about partnerships. We have several partnerships with not only vendors, but also other core facilities within McMaster. And we do this because this allows us to not only get more educational material from the vendors, it also allows us, the staff at COM, to actually get trained on specific techniques that might be coming to the market very soon. So we basically profit from those partnerships with the vendors. They also support outreach and educational activities. So next year, we are going to organize our first summer school, which will be a week intensive workshop with theory and practical. Again, details will be announced later towards the end of the year. We also have the opportunity to now have speaker series, thanks to the BIMR. Thank you for that. And again, we also might consider in the future to have our own seminar series, if people are not too overloaded, but that's something that we would like to consider. And again, this also allows us to get in contact with the vendors and the prototypes that they might be coming up with. And this might open new research avenues here at McMaster. And again, we can be then at the forefront of science worldwide. And also, as I said, priority is technical support. So we expect that if one system has to go down, it will be repaired as soon as possible so that your work is not affected and you can actually keep producing data. As an example from CCM, we have now created a workflow, or we are starting to create a workflow for cryo, correlative light microscopy, electron microscopy imaging. So we obtained the instrument very recently. And we also share the imaging space, the workstation space with CCM. So it's a very nice area where you basically can exchange ideas with electron microscopists and light microscopists. And we are also organizing joint image analysis workshops, which you can find on the CCM YouTube website, where we, on my part, we discuss open source software like Fiji for your image analysis needs. And on the CCM side, they can actually train you on Jupyter Notebook and how you can write code to tackle the same issues that we might be facing in light microscopes. We're also part of organizing different societies, the Microscopy Society of Canada, the Nano Ontario, which is going to happen in the 17th of November. So mark your dates. Next week, there'll be a tour at COM, which will be quite interesting. And the talks will be also at the CIBC Hall. And in the CCMA, we're also going to have host a session there on image analysis needs, or basically the cutting edge technology that's been developed recently on open source software. Yeah, pretty much. And again, the partnership with the cryo is also with Zeiss and Physics. And again, as I said, we have received the LSM confocal with the NeriScan very recently. And this machine is actually quite interesting for some of the people that I can see already in the audience. Looking at Cecile there. We also have the BIMR collaboration. So we are proud. So we are proud to establish a seed grant program. So this is an idea to ease the work of starting PIs that might not have the necessary funds to produce data or to use our facility as long as they wish. So the idea is that you can apply to these grant programs. You receive a monetary support up to $2,000. And this can actually get you started on acquiring images for your grant proposals and grant submissions. The applications are reviewed on a rolling basis. So I think we just got our first approval very recently for three seed grants. And again, early PIs are really encouraged to apply for this because it really can make a difference in your applications for funding. I think if you scan the QR code, you might be able to go to the webpage. Again, all of this is on our website if you want to get more information. So we also hosted initiatives at COM. As some of you might have known, last year we organized the C2 Discover image competition. We asked our users to submit their best images, the most artistic and also scientifically viable or purposeful images that they could master. And these are our winners. So we have basically we have basically the first prize on the biological sample. Second one was, I think, these channels that Boyang Zhang is actually doing on the second floor. And the third one was basically DNA and protein by AFM, which is a technique that most material people actually are using it downstairs. Yeah, this was quite successful. We actually had a lot of positive feedback. A lot of people voted. It was really good to engage the community and actually get them started. And this will be also happening next year. So all of you that are in the audience there are making images and you are proud of them. Don't forget that you can apply for this and you do get a nice prize also. Moving forward. So we want to strengthen different strategic areas within McMaster. We want to dynamize the advanced materials and manufacturing. So we are, once the macrofabrication lab is operational, we will be able to do light activated 3D printing. We also are interested in soft materials. We want to characterize polymers and biomaterials, biosensors. We also are developing correlative imaging approaches, as I said, in the cryo-electron EM microscopy. We also have projects regarding aging across the lifespan with multiplexing imaging. We also have, we also want to address the growing burden of chronic disease, again, by multiplexing imaging of phenotypic markers. And there's also projects regarding infectious diseases and how to tackle them. So this is a quick flavor. There are many more things that we can do, but these are basically the focus that we are focused on at the moment. Okay, so let's now start with the eye candy, the interesting parts, what people are doing in the facility. These are just examples of things that material scientists, biologists, engineers have been doing and tinkering in the facility downstairs. So the first project that we had, and this is actually one of the first people that contacted us, really back in, I would say, September 2021, it was Ryan. He had an issue that he had these membrane filters that he would put on a stream of water, and he was basically quantifying the filtering of microplastics. So these small green particles that you might see on the left are small particles of microplastic. So this is basically the face of the filter that was facing the water flow, and this is the back side where you don't see much. And what we did, we just did a typical, let's just image a volume, the volume of the filter. And let me see if this is working. Hello. Okay. Oh, there it is. Sorry. So again, we are doing a volume imaging. So we're going through the whole filter with a high resolution, and we can see that we have the particles there. And when we do a cross-section profile, we see that the particles get retained really at the top part of the filter. They don't really have the size, the small size that actually allows them to penetrate through the porosity of this filter. So again, simple project, simple question to tackle. The imaging was quite easy to do, and actually he was very happy with the results. We've also been imaging, and this is work done by Djadranov and the Jose Moran Mirabal labs. We've also been imaging film-embedded lipid nanoparticles. So again, here we have a film with these lipid particles, which are highlighted there. The color represents the depth of the Z where they are located. And you can see that this this is a 65 by 100 micron volume by 100 was imaged relatively quite fast. And you can get an idea about the different size of these particles, and you can actually get an idea about the distribution along the apical basal axis. One thing that they've also decided to do was to dissolve these filters for vaccine delivery. So again, this is exactly the same imaging that we did before, but now we're doing actually a time lapse. So we're going to be imaging the volume over time. So we had a data dimension there. And while we're imaging, we're going to add a droplet of water. Again, Mohamed, correct me if I'm wrong, but you add a droplet of water that's going to dissolve the filter. And then you can see basically how these particles are going to react to that dissolution. As you can see, this was imaged at 0.27 frames per second. So relatively quite fast for a big volume like this one. And if the movie start playing, there we go. So again, there's a bit of a movement all around. But if you look into the bottom video, and again, the tracking was done for these particles, and we observed that the orange particles, which are really at the base of the filter, they actually were dissolving faster than the ones on the top, which tells us that basically where the drop water contacted the filter was actually where the dissolution was happening faster. And yeah, quantification was it's not there, but again, trust me on this one. Yes. So some of the things that we also do at COM are related to AFM. Excuse me. With AFM, which is atomic force microscopy, we can assess DNA protein interactions as an example. And this work was done by Monica from the Andres lab at FHS. So the idea is that she coated the surface of her, I think it's a mica, of her mica. She coated it with DNA, and she basically also put some proteins which are unspecified or they don't matter. So basically, she actually then used the AFM to see how these proteins would interact with the DNA. And one thing that she got were these images, which actually got her a prize in our image competition. And if you look into the right image, you have now a height map of this structure. So you can see basically the DNA over there. And then the big proteins are the bigger peaks that are popping out. So it's a 1.3 nanometer thing. And one thing that we can do in the facility is actually we can also 3D print these structures. So again, you can make your artistic photos touch. You can touch the DNA, touch the proteins. And for science communication, this is fantastic. When you have little kids coming over and trying to explain what DNA and proteins do, you just show them this. They can touch literally the DNA, touch the proteins, and can understand what the AFM is doing. So it's really, really cool things that can be done with these techniques. Another thing that we can do with AFM is regarding nanocellular structures. So this work was done in the Moran Mirabal lab by Mohamed, who is now working for COM, and we're very lucky to have him there. So basically what he did was he 3D printed these PEGDA structures, and he coated them with the nanocellulose nanocrystals. So the image on the left, which looks like a bit like a hill on the middle of the plains, that's basically a PEGDA structure. And if you look, so this is a 10 micron by 10 micron image. If you look in high resolution, you can see that it's all coated in those yellow nanocellulose particles. So you can just get an idea about the dimension of things that we're discussing here. This image actually took, if I'm not correct, if I'm not mistaken, 2 hours to just acquire that image. But it's fantastic, just amazing that you can go from this 10 micron by 10 micron down to the nanometer level in such a way. On the right side of this image, we have now the nanofibrils, the cellulose that they work on. And this is just a fantastic image that shows how the breadth and of these particles of these cellulose is actually arranged, and the resolutions that these fibrils are actually containing. So we're talking roughly 10 nanometer thickness fibers, and then they actually branch together and they create thicker and thicker and thicker. And this is basically what this image is trying to represent. Fantastic work. I'm a biologist, so I always like to put some biology. I know that the audience might not be too excited about it, but we can do really cool stuff also with our OCT. So let me explain what happened here. So we have users that are working on Drosophila larvae. They are putting the larvae on different diets, and they want to see how the heart is going to react, how the heartbeat frequency is going to react to these different diets. So the user approached us. We trained on the machine. She started making the movies. But if you look into the movie, and I know it's a bit difficult to see, but because of the way the larvae breathes, the heart is actually moving or drifting a bit in X, Y, Z. If I make the movie faster, so let me see, can I interact? I cannot. The presenter model doesn't allow me. But basically, trust me on this one. If I wanted to register that, make basically a camograph of that heart, there'll be way too much drift, and I cannot actually get a clear idea about the frequency of the heart rate. So one thing that we could do is actually we can write a macro for Rachel. And what we did was we now registered the whole movement of the larvae on the trachea, which is adjacent. So it's located in this small area here. And the trachea is not moving at all because basically the trachea is very static in the Drosophila. And when we do that, now you see that there's a lot of G3 because this is actually the compensation of the movement that the heart was suffering before. And now the heart is basically very, very static. It's roughly in the same spot. And when we do the camograph, which is basically a reslicing of time and your x-axis. So basically, this is basically the x-axis over time. You can now see that now I can register the peaks of which this heartbeat is actually beating. And this allows me to calculate the frequency in a much nicer way. So she was very happy because she doesn't have to do duration manually, and she could actually get proper data out of these experiments. Other things that we do in the facility are regarding, for example, bone imaging. So we were approached by people from Valiant Research Group. They had a very unique, they do photoacoustics, and the dye that they use for photoacoustics is actually fluorescent in the near infrared part of the spectrum. So we told them, well, unfortunately, we don't have such a detector, but we can try to use the far red detector to actually see what we can see. And we managed to get basically this reconstruction of the bone of the mouse, if I'm not mistaken. And again, you can even see some cells highlighted by the fluorescence. And this was the best we could offer at the time. So it's not, I mean, there's a lot of background still. But now with the new instrumentation that we have, we can actually do this because we now have detectors for the near infrared spectrum. And we're really looking forward to image these samples again with the proper instrumentation. One thing that I forgot to mention before on the AFM is that we are also going to get upgrades on the AFM very soon. We can already do QNM. We can do conductivity measurements. But soon we'll also be able to do fluid measurements once we get the fluid chamber in the facility. So if you want to measure the thickness or the stiffness of your hydrogels, they'll be possible very, very soon. Yes, that's all I had to say about this slide. Again, we can also do organoids. So again, this is just, oh, sorry. Is it moving? Sorry, let me just start again the slide. I apologize. Yes, it's not working. So we were approached by Sarah. She wanted to be trained on one of our systems to image her organoids. So these are organoids which are mounted on a matrigel. And her question is, I want to see the distribution of my cells labeled with a marker in Psi5, which are the cyan cells there. And I want to see how they actually where they are located inside my organoid. And what we did was did volumetric imaging of these organoids in high resolution, X, Y, and Z. And what we can do in the end, we can actually make these animations which show you the volume of the organoid. And you can actually see where your cells are exactly located. And hopefully you can even measure distance between the cells and their location relative to a fiduciary point in space. So again, really cool stuff. And again, yeah, really interesting things to do in the facility. One thing that we also do is that we have super resolution systems, and the super resolution system that uses or gives the best resolution is Storm. So this is work from Mohamed. So again, this is a cellulose nanofibril that we saw before in the beginning. What we did is that we put this sample in a specific buffer, which basically makes the fluorophores inside the sample unstable, and they start blinking on and off very, very rapidly. And one thing that you can do is that once they start blinking, as you can see here, you can now start registering these events over time. And through some algorithms, some mathematical algorithms, you can then actually pinpoint their exact location with much more accuracy than you would with a normal imaging. And in the end, what you get, you start from this image, which is okay. But after processing this image, you get something like this. And now you can actually see things that you cannot see before. You can see patterns that were hidden because of the imaging that you were doing. So Storm can actually expand your eye into these things that might be hidden by just not using the right technique. And it's, again, very, very cool things that can be achieved with this. Again, one thing that you can do is that you can then measure the thickness of these structures. And one thing that we can see here, there's a bit of coiling highlighted there by the little dotted line. And when you measure the distance between the coils, you see that we're measuring a distance of 50 nanometers. So really, really, really high resolved. Again, I might have missed some things, Mohamed, but I apologize, but that's it. Again, we can also do this for LNPs, for the lipid nanoparticles. And what happens there, we just apply the Storm technique. And now instead of going for a 200 nanometer image, we can actually see that they are actually smaller than that, around 80 nanometers. And again, this was a result that was also shown by AFM and by DLS. So again, it just strengthens the point that you want to make, and the reviewers will be happy with that when you publish the paper. And again, because of some shameful self-advertising, there'll be a seminar next week on super resolution techniques. So we had the lucky acceptance by Pedro Pereira. He was a postdoc at the Henrique's lab in London, where they develop really cool techniques on super resolution. And he kindly agreed to actually host the next seminar. This will be a virtual seminar, and I highly encourage you to attend it. He'll be talking about super resolution, 3D printing, bioprinting, bioinks, really cool stuff, and I'm really looking forward to see this talk. So again, don't miss it, next Friday at 2:30 on Zoom. Yes, on Zoom. What else can we do? We can also do IBEX. So again, this is something that, again, we can do now, yes, with the area scan. We'll be able to do much better next year once you get the full system for this. So just to give an idea, so before we're talking micrometers, this is a whole piece of lung. So the scale bar there is 1 millimeter. There's not a mistake there. So this is a very, very big chunk of tissue. And what we did here is that we image the spectral profile of this lung tissue. And why is that? Because we had several fluorophores in there. We had up to 8 fluorophores. So the lab stained, Megan stained several fluorophores inside the sample. And now, because of the possibility of doing spectral analysis, she can see the profile of the spectrum and actually pinpoint the areas of interest for her, and actually can separate all the channels at the same time. So what she can do is she can then see, okay, I can see the staining in blue, like in the blue range of the spectrum. If I look into the green part of the spectrum, I have a different type of marker. If I go into the red part of the spectrum, I have a different type of marker. If I go into the, I guess this is Psi5, into a different range, near infrared, a different range. And when I put everything together, I get something like this. So again, for immunologists, this is extremely important because they can actually label different markers with different fluorophores. And then in a simple way, they can actually extract all the information at once, and then they rinse and repeat. They do chemical bleaching, they do another round of staining, and they get more proteins localized where they want to see them. So really interesting stuff. One thing that they also do in the neighboring lab, at the Koenig, well, Jordana lab, but soon to be Koenig lab, they also image the mouse intestine. So this is the full intestine of the mouse. They call this a Swiss roll. Again, you can have an idea that this is a very, very long piece of tissue. And again, we image this on the spectrum. We acquire 34 spectra, and then we separate them and we mix them. And in the end, we get the 7 proteins located very uniquely within the structures of the intestine. Again, more self-advertising. On the 25th of November, there'll be Joshua Koenig that I just showed before. He'll be talking about his strategies and his ideas for his lab. And again, I'm really looking forward to this talk. It's going to be really, really interesting. He'll be showing us really, really interesting data. Big data, but again, very interesting results that can come out of these techniques. So we also are imaging 3D printed scaffolds. Excuse me. So this is work done by Moran Mirabal and also Angelo Accardo. So they are collaborating on these. So if I understood correctly, they are 3D printing these scaffolds, and then they coat them with a Psi5 cellulose nanocrystals. Again, the crystals that we saw before for the AFM, now they are labeled with fluorescence. And when you image them, you can see the structures that you 3D printed very, very nicely with fluorescence on the left. But if you look on the right, this is just a 3D printed scaffold. There's no fluorescence coating. And what we did here is actually use the reflected light to actually get an image. And we can see the patterns in this scaffold. Let me just play that video again. And you can see the scaffolds and the structures that are 3D printed. And you can see if there are inaccuracies with your 3D printer, you can assess if everything is working properly and moving on to the next part of the project. And again, this is not naive. There'll be a talk on December 2nd by Angelo Accardo. We are fortunate enough to have him around. He's coming, right, Jose? He'll be here, and he'll be talking about what they do in his lab, and again, 3D printing in the bio environment. Yeah, cell mechanobiology, so expect some biophysics and some interesting stuff on the 3D printing world. So what else can we do that I could not show you? Many, many things. Basically, you name it. We can image biofilm thickness using AFM or CT. We have some people that have starting to do that at the moment in the facility. Again, our OCT has a resolution of 3 micrometers. So if you have something thinner than that, you have to go for the AFM. If not, OCT is the way to go. Again, you can also do some live imaging. That's something that we still haven't started really. There's not been a lot of requests for that, but this is something we can do in all of our systems. Our confocals are equipped with imaging chambers that can regulate temperature and CO2. We can also assess pH with fluorescent dyes. This is something that's going to happen next week on our area scan. We can also do SIM, structural dimension microscopy, and live imaging of organelles. This will be done next week from Cecile Fraden's lab. We also have now techniques, the area scan that I didn't mention too much. I just mentioned in passing, but this allows you to image not at the super resolution level, but pretty close to that. We call this extended resolution techniques. We also will be having photon counting systems very, very soon, which can lead to FCS and RICS, and many, many other things that it's hard to put in one slide. So again, if you have any scientific question that you want to address and you think we might help, please do so. We are there for you. We are here to support your research. Do not hesitate. Don't be shy. And be critical. If you feel that you need something that we cannot offer, request it, complain. We like complaints because then we can raise to our directors and push for the instrumentation to come over. For more information, you can go to our website, com.mcmaster.ca. Any questions that you want to address, send them to me at our common email at com@mcmaster.ca. If you want to bug our director, there's his email over there also. And again, just a quick reminder for advertising. Don't forget the seminars that are coming. 18th, super resolution from Pedro. 25th, Joshua about IBEX. And on the 2nd, we'll have Angelo Accardo talking about 3D printing and biomechanics. And thank you very much for your attention. I'm open to questions.
Any questions?
Is there any talks about being able to image BSL2 level
We can image BSL2 as long as the receptacles where they are located are properly sealed, and we have small volumes of imaging. If you want to image liters of something, we cannot do that. But if you actually enclose them in a small petri dish, properly sealed, a glass bottom, hopefully, again, we can advise you on that. You're more than welcome to do so. So again, as long as everything is sealed and in small volumes, we can image them in our microscopes. Come talk, ask us, and we'll see. We can work with you on that. That was another question, I think. Sorry? It's a matter of scale. So with light microscopy, we always are conditioned by the diffraction limit of light. So Abbe's equation tells us that you cannot really go beyond 200 nanometer resolution because of the waves of light interfere with one another. There are ways to circumvent that, which are the super resolution techniques that I mentioned. So SIM has a way to figure out to work around it. Storm has also a different way to work around these limitations. STED has another way to do so. But again, in light microscopy, you always don't expect to be able to go beyond 20 nanometer resolution. There's min flux. I know that I don't even need to look there. I know there's a min flux look at there, and that will give you like 1 nanometer resolution. But that's basically a technique that just started, I would say 3-4 years ago. And yeah, I mean, I guess min flux can be the transition between EM and light microscopy, just a matter of scale. But I think we still need to see if actually it's a technique that has potential and is going to be having a lot of applications. But we might have it in the facility sometime. Who knows? Fingers crossed.
For electron microscopy or just like normal white light microscopy, everything's reflecting light at the electrons, because you don't get that specificity or contrast as well as super fluorescence.
Any other questions? Yes, Cecil. So at the moment we have CACO2s, we have COS1 and COS7 cells, we have RAWs, we have 3T3s, we have what's the A? There's an A5, macroscopic cells. Yes, most of them are, yeah, all of them are eukaryotic, yes. Some of them are. So we do have a line which is GFP, which we use in our trainings for live imaging, and also for our demos. but it's just like normal GFP. It's not labeled anything. We do have reagents in-house for local consumption between the facility that would allow us to stain for certain components of the cells, like phalloidin, SIR for microtubules, and the mitochondria. But yes, so out of our cell lines, only one of them is actually naturally expressing GFP. But again, we can always help you transfect the cells with markers for expression of fluorescent markers. That's a possibility. We have protocols, SOPs already in place. So we had something I forgot to mention. We had this summer a summer student whose role was actually to put in place the SOPs for the cell culture. So we have established SOPs for staining, for freezing cells, for keeping the cells, and so on and so forth. Yes. Any other arms?
Well,
Yes, Alex.
with the new IR fluorescence microscopy, what are the wavelength ranges for?
So on the IR, we can go, I saw it yesterday, we can go up to 950 nanometers. Sorry? 920? Sorry, 920. I apologize. I was giving you 30 extra nanometers. 920, yes. The ultimate resolution. So it has an area scan. So you could go, Zeiss is going to tell you they can go 90 nanometers. I would say we can go 120 on a standard base. If you have an amazing sample, we might always go beyond that. But like for normal preparations and stuff, we can go up easily to 120, 150, no problem with the area scan. And it's actually extremely fast. So if you also want speed, you can change the sensor to act not only as a sensitive sensor, but also to rasterize 8 lines at a time. So that allows you to also increase the speed. If you want to do calcium imaging, that's a system to go for. You can actually achieve very, very high imaging speeds with the area scan.
In terms of qualitative microscopy, I know you're working on light and electron. Is there any chance to do light and AFM?
Yes, that'll be possible. If you have the fiduciaries, we'll be able to do so, yeah.
Actually, it's better.
And we've done it before, so we have already the expertise in that. So the cryo is something that we're going to start now with AEM. We realize that there are some things that we still need to get to fully establish the workflow, but it's something that we need to check with CCM what's going to happen. But yes.
Liquid cell for the AFMs.
I hope so. Thank you very much for the opportunity. Appreciate it. Thank you, everyone.